what nanobodies can do for structural biology. There's a lot of new faces in the audience, so I think it's useful. As you know, many proteins, also many drug targets, are in fact what I call moving targets. These are moving proteins. And for the last 10 years, to me, my lab has essentially tried to build what I call a molecular, it's a very difficult word, zoopraxiscope, but it essentially deals with, if you look at a moving target, try to trap as much as possible pictures of stills of that moving target to reconstruct your moving target and to understand its function. You will immediately understand why I use a camera as an example. This thing doesn't really work. What we essentially do is we are not using conventional antibodies built from heavy chains and light chains, but we use nanobodies derived from heavy chain only antibodies that occur in camels. This is in fact a discovery made in Brussels about 30 years ago, and we found out that in fact these nanobodies, these single domain antibodies, are very useful in structural biology because they bind conformational epitopes. So you can use them to lock conformations of proteins, and that's indeed the feature we use systematically to look at dynamic objects. And I will start and illustrate that with older data we essentially obtained with Brian Kobilka, and these were studies on G protein coupled receptors. A very short introduction: it's a moving target, and the receptor alone can trap some G protein to cause what we call basal activity. And then you can use small molecules, agonists, to in fact stabilize what we call active conformations that better bind G protein and signal more, ending up in full agonists or partial agonists. And of course, you can also identify what we call inverse agonists that block the receptor in an inactive conformation and kill the cellular signal. And until 10 years ago, we knew that these conformational changes were occurring, but we had no clue how these conformations were looking at. And Elsa and myself 10 years ago set out to make nanobodies that lock these G protein coupled receptors in different functional conformations. And the first experiments were on the beta-2-adrenergic receptor, where indeed we identified nanobodies that lock this receptor in an inactive state, and we know that this is an inactive state because an inverse agonist is bound. We also selected other nanobodies that stabilize the same receptor in an active state, and we know that this is an active state because an agonist is bound. And over the years we did this exercise for several different G protein coupled receptors to finally come to a kind of a general scheme of GPCR activation. And this is in fact what is now believed most GPCRs go through when you go from an active state in green to an inactive state in gray. Yes, it's essentially a conformational change where you grab an agonist that is smaller and you open up a cavity for the G protein to bind. And whenever we look at these active state structures versus inactive state structures of G protein coupled receptors, we see the same phenomenon. So this is a very clear illustration of how you can use nanobodies to lock proteins in unique conformations to understand their function. Later on, we moved to protein complexes, and I will come back to this later, but we did not only immunize with the beta-2-adrenergic receptor alone, but at a certain moment we thought, why don't we look at the whole complex? And we start to immunize with the beta-2-adrenergic receptor, cross-linked, in fact, to the G protein, trying to select antibodies that bind this complex, and by binding to this complex, just by mass action, stabilize this complex. And this is a very famous nanobody. This is nanobody 35. You see the nanobody here. This is the GPCR. This is G alpha, G beta, G gamma. And you really see that indeed it binds an overlapping epitope here in the nucleotide-free version of the G protein, thus stabilizing the complex. And by solving this complex, Brian indeed managed to finally explain how an agonist that binds the outside of the receptor, activates the receptor, and you see indeed the opening here at the inside and the G protein that binds this cavity, opening up the nucleotide binding site, allowing GTP to leave this active site. So again, conformation, linking to structure by using the unique properties of nanobodies to stabilize conformational epitopes. What's really nice is that these nanobodies are small, but you can also express them inside cells. And this is just an illustration of this thing. So what you see is 3 times the same cell, yes, stained, looked at in different colors. Here you see green, and that's the nanobody that binds the active state of the beta-2-adrenergic receptor fused to GFP. So you see in this case that the nanobody is throughout the cell. The GPCR, the beta-2-adrenergic receptor, is colored in red, and this is just the overlay. And what I will do is I will just add adrenaline, I will add the agonist to the system, yes, and then you see biology in real life. So at time 0, we add the adrenaline, and what you will see is that the nanobody is recruited to the membranes that turn into an active state. The GPCR turns into an active state, recruits the nanobody, and at the end you will even see that these GPCRs in the active state start to accumulate in endosomes. So it's really a tool that allows you to translate structural data obtained by X-ray, obtained by cryo-EM, into a cellular context, into biology. So nanobodies are small, but we are entering the cryo-EM era, and small is beautiful, but in cryo you want your things to be big. Yes, so we thought, how can we turn our small nanobodies in larger but rigid units, retaining all the nice properties of these nanobodies. And the trick we applied is, in fact, so this is a nanobody, and what we did is we essentially cut open one loop, one 3D conserved loop in the nanobody, and then we take another large protein where we also cut open a loop, yes, to make this fusion that we now call a nanobody that fuses, a megabody, sorry, that fuses the nanobody to a large protein in a rigid fashion, because this involves 2 peptide bonds. And it turned out that we can do this. So these are crystal structures of nanobodies fused to different scaffolds. This is 70 kilodalton, this is 100 kilodalton. You can have them in different shapes and different tastes. And this is now a tool that is visible under the microscope. So, and that's essentially what we wanted to obtain. And it's in fact, these are the first data of using these megabodies now in cryo-EM. I mean, this is the GABA receptor, and you know there is 2 alpha subunits, 2 beta subunits, and 1 gamma subunit, but they are very similar, so it's almost impossible to align projections of this thing, except if you have a new unique feature. And we made a nanobody that binds only one side on this pentameric system, turning into a megabody, and you see the megabody there, allowing you to align these particles and to refine this structure. What we also found is that in many cases it resolves the preferential orientation issue. Yes, this is again the GABA receptor alone, and you see that most of the particles are, yes, top down, like we call it, and only a small fraction is on the side. If you add a megabody to this system, you tumble your particles, and you end up with a more even distribution. So adding something to your protein does not only make it bigger, gives it new features to align your images, but it also, in several cases, we have seen solve preferential orientation. And these are the net results of all this work. Yes, really high resolution structures, and where we for the first time see how Valium or Xanax binds to this GABA receptor, because it's amazing to see that we are taking these drugs for 20, 30 years now, but nobody ever has seen the structures of these ligands bound to this ion channel. Okay, this is another application where we use these megabodies as fiducial markers. If you take a small transporter and you put it in a detergent, you essentially get a blob. Yes, and it's impossible to, again, align these blobs, but by adding a megabody, so you have the blob there and you have a megabody binding to the protein, this allows you again to align these blobs and to solve the structure of, in this case, a liver transporter for bile acids. Yes, and if you use different megabodies built from different nanobodies that bind different conformations of the same transporter, you can even solve structures, different conformations of this particular transporter. Brings us to another application. I mean, we discussed purification of proteins yesterday. We thought, why don't we use these nanobodies to purify proteins? And in fact, I think we did it in a smart way, because whenever we select nanobodies, we identify what we call nanobody families, where some have lower affinity and others have higher affinities for the same epitope. And what we essentially do is we use a low affinity binder as what we call a trapper to trap a protein on a bead, yes, and we use a higher affinity nanobody to elute it again. And that means that we have a very selective way to trap proteins. Our elution again is selective, and on top of that, yes, you trap your protein with a conformational nanobody, but you also elute your protein with a conformational nanobody. So that means that you only elute well-folded protein. And we use this, for example, to just pull out the adrenogen receptor from a cellular culture. Yes, we just GFP fuse the protein, and then you see that you can pull out the complex of this receptor with all its cofactors as it is described in literature. So it brings me to the last and the most recent application we are focusing on, and that deals with protein-protein interactions. I mean, this is a very naive scheme, but it's crucial in the sense that whenever proteins associate, there is conformational changes again, and if you can trap the conformational changes that are associated with the formation of your complex, you can make nanobodies that stabilize this complex. Yes? And we are really focusing now on the stabilization of transient complexes, using nanobodies aiming to solve structures and to study the biology of these complexes. I already gave an example. Yes, this is the nanobody 35 stabilizing this complex. We managed to stabilize complexes between a kinase and ubiquitin. Yes, so this is an enzyme substrate complex that we stabilized with a nanobody. And I want to end this story with some recent work on KRAS and its nucleotide exchange. Sorry, this is KRAS and this is SOS1, the nucleotide exchange factor, where we started to play with to really illustrate that you can use nanobodies to stabilize these complexes in an allosteric fashion. So the tricks we use is simple. We cross-link our complexes before we immunize to make sure that we present epitopes that are characteristic for the complex to the animal, and we also devise fancy tricks to pull out binders that specifically bind the complex. This is just one of our tricks. So this is yeast display, where you put a nanobody on one nanobody in 50,000 copies on a yeast cell. And what you do, in fact, if your complex consists of two proteins, you give it different fluorescent dyes. So protein A is blue, protein B is green, and then there is three options. Your nanobody binds protein A, so it means that your yeast cell is blue. Your nanobody binds protein B, means that your yeast cell is green, or your nanobody binds the complex, and then you have the two colors. And you can sort these yeast cells just by facts. So we are using these displayed by FACS to pull out binders that stabilize the complex, that bind to protein A, that bind to protein B, or that bind nothing at all.